Welcome back to Epistem, and today the topic of our discussion is vaccine, their types and the preparation. So what are vaccines? Now vaccines are actually a suspension of live or inactivated microbes, their fractions or genetic material, and vaccination is the process of administration of these vaccines in the living animals in order to promote immunity and prevent any type of infectious disease. And without the risk of a full-blown infection, vaccines assist the body in learning how to defend itself against the disease. So here we actually face a trailer of a disease, not the entire disease. A vaccine's immunological reaction may result in fatigue and discomfort for 1 or 2 days, but the subsequent protection may last a lifetime. So we have seen whenever we took the COVID-19 vaccine or the tetanus vaccine, so we got sick for 1 or 2 days and we had like pain in our hand where we took the injection like that. So this is the time period when the body means the immune system of our body prepares the antibodies against the antigens or the vaccine that is introduced in our body. Now, in order to activate the body's defense mechanisms, the vaccines imitate an infection, the presence of a disease-causing organism in the body. So the vaccines actually imitate an infection. They don't cause a real infection, and there is only some symptoms and some like symptoms we just see of the disease and not the entire disease we have to face. An antigen, also known as the molecule that triggers the immune system to start making of the antibodies. They serve as the active component in every vaccine. Now antigen, we all know that in response to an antigen, antibodies are produced, and these antigens, they are actually the main component of every vaccines. So along with the preservatives and other ingredients needed for the formulation, so the main part is this antigen. This is the key ingredient of a vaccine. So these antigens are actually this genetic material, the fractions, the inactivated microbes. So they are the antigens. Now here we have a simple representation of how a vaccine works in our body. So we introduce the dead or weakened bacteria in the body by injection or through orally, depending on the type of the vaccine. And next, what happens once they are introduced, the white blood cells, they get activated and they produce the antibodies specific for this antigen. And next time when the real infection will occur, the real bacteria or the virus, they will attack our body, these antibodies and also the memory cells that were created, they will prevent the infection. Now here are some criteria for a good vaccine. So firstly, it should be safe for the immunocompromised people, because vaccine is a universal thing and it can be like used by all the persons that are present in the world. And vaccination results in the complete prevention of infection, which is the ultimate aim for any good vaccine. The immunity should be long lived so that we don't have to take another dose within 1 or 2 weeks. Now, particularly for the use in tropical countries, the ideal vaccine should have high thermal stability. To save further on the administration cost, a single vaccine might be multivalent. Now, multivalent means that one vaccine has the ability to prevent multiple diseases, like the DPT vaccine. So DPT vaccine stands for diphtheria, pertussis and tetanus. So this one vaccine can prevent 3 diseases. Now, many vaccines are administered parenterally, which adds to the total cost because of the need for skilled personnel. Like skilled personnel means nurses, doctors. So we cannot, like a normal person cannot like give an injection to another person. It is difficult for them. So that's why you always trust a nurse or a doctor for that. And for this reason, the price of the vaccine increases. Now, administration via mucosal surface and preferably orally offers many advantages for vaccine delivery. Next, we have the types of vaccine. So we have like 7 types of vaccine and we will learn each one of them in this video. First one is live attenuated vaccine. So live attenuated vaccines, they are prepared by using the living pathogens and they are weakened. Now this weakening is done by the repeated culturing And the live attenuated vaccines are created by choosing the types of bacteria or the viruses that still elicit a strong immune response but do not cause the disease. So these specific bacteria or virus that we are using while we are making the live attenuated vaccine, so they have the ability to turn on our immune response, and but they do not cause the disease. It just turns on our immune response, produce the antibodies, that's it. Now the examples of live attenuated vaccines are measles, rubella, smallpox and chickenpox vaccine. So here is the illustration of how the live attenuated vaccines work, and the upcoming all the illustrations are taken from Pfizer. So here we introduce the vaccine in our body, and these are the live viruses. So they will come and they will enter the human cell, and in our human cell the cell machinery will help to read this genetic code and form the viral proteins, and which will help to produce the viral particles. So these are the newly formed replicated weakened viruses. Now these viruses are actually detected as a foreign particle by our body. So the antigen presenting cells, the APC cells, which are like the examples are the macrophages, the dendritic cells. So this type of cells, they will engulf those foreign particles. Like this is the weakened virus here. So they will engulf them and chop them, and these antigens will be presented by the APC cells. And these antigens will be detected by the T helper cells. And then the T helper cells, they release the cytokines. These cytokines help to trigger the B cells to produce the antibodies, and also the memory cells are also produced. And when the real infection will occur by the introduction of the full-blown, full potential viruses, so they will be recognized and they will be destroyed. Next we have the inactivated vaccines. Now inactivated vaccines, for making these vaccines, we take the live pathogen and we inactivate them or kill them so that they can't replicate. And they are prepared, like the killing of these pathogens are done by using the gamma rays or some chemicals. Examples are polio, hepatitis A and rabies vaccine. So we introduce these vaccine in our body, and since they are dead, so they don't have the ability to replicate. So that's why they are directly identified by the APC cells, and once they are engulfed by these APC cells, they are chopped off, and the antigens are presented by the APC cell. The T helper cells next identifies it and release the cytokines, and then the B cells produce the antibodies. Now here we see that the antibody levels fade with time, and usually for this type of vaccine we need to take additional shots. For, like we have seen, like for polio vaccine, we have seen that the infants, like the 2 months old, these 2-month-old infants take the first vaccine of polio, and then after like 2 months, that is when they are 4 months, they take another shot, like this. Next is the subunit vaccine. So subunit vaccine are prepared from the piece of microorganism, and not the whole microorganism is used. So examples are polysaccharide vaccine, conjugate vaccine and protein vaccines. And examples are influenza and pneumococcal vaccines. So at first we will talk about the polysaccharide and conjugate vaccine. So some bacteria, they have the ability to hide from our immune system by developing some polysaccharide chains on their surface, like these red ones. And in laboratory we can tag these polysaccharide chains with some proteins which help the immune system to identify them. So this preparation is introduced in our body. Now the APC cells, they identify these foreign particles and they engulf them. Now the antigenic part, that is this polysaccharide part, is presented by this APC cell, and next the T helper cell, they identify it, and the cytokines are released and the B cells produce the antibodies. So this is how the polysaccharide and conjugate vaccines work. And next we have the protein vaccine. So in protein vaccine, so we actually work with only the viral protein, not the entire virus, and we tag this viral protein with the adjuvant. Now adjuvants, they act as like enhancers for our immune system. So they are tagged with these adjuvants, and next they are taken in by the APC cells, and the antigen is presented by them, and the T helper cells, they identify this antigen and release the cytokines, and the B cells produce the antibodies. So this is how they work. Next we have the recombinant vector vaccine. So here what happens, so specific genes responsible for a particular type of key antigen can be introduced in the attenuated virus or the bacteria which act as the vector. So here we use weak bacteria or virus, and they act as vectors, and inside this bacteria or virus we introduce a specific gene that codes for a specific antigen. So here is where is the real virus, the real one. So, and these are the red ones are the spike proteins, which is coded by a certain segment of this gene, of this genome actually. Now we separate out a specific segment of this gene and introduce it into the weakened virus. So you can see these spike proteins, they are the same shape, but they're actually blue because they are not like potential, they're not highly potential to cause the disease. So that's why it is differentiated from this one by giving the blue color. Now they're introduced into the body. Now since they are the live cells, they're weakened but they're live, remember that, and as they are introduced into the body, the APC cells, they take them in, and the genetic code is released, and the cell's machinery help to produce the viral protein. Now this viral protein is a foreign particle, and they will be presented by the cells, and the T helper cells, they detect it, and similar way the antibodies are produced. Next we have the toxoid vaccine. So in the toxoid vaccine we actually use the inactivated bacterial toxins. So examples are diphtheria and tetanus toxoid. So we introduce them in the body, and remember that we tag these toxoids with the adjuvants, because adjuvants, they enhance the immune response. We already know that. So once they are prepared, they are introduced into our body, and they are directly taken in by the APC cells, and the antigens are separated out from the adjuvant, and they are presented by the APC cells, and the T helper cells, they detect it, and the B cells, they produce the antibodies. The same way the antibodies will be produced. Now when the bacteria, they attack our body, like when the bacteria will attack, so they will release these toxins, and these toxins will be identified by the APC cells, and these antibodies will attack these toxins and prevent our body from any type of infection. Next we have the DNA vaccine. So the DNA vaccine, here what happens, we introduce or inject the DNA directly into the muscle of the recipient, and the host cell, they take up the DNA, and they produce the proteins, the immunogenic proteins, they are produced. So ZyCoV-D is the world's first and India's indigenously developed DNA based vaccine for COVID-19. It's an example. So it's a very simple thing we just learned, like we take the plasmid DNA of the virus, and then plasmid DNA from the virus or the bacteria, anything, and we introduce it into the muscle of the person, and then the host cell will produce the viral protein or the necessary antigens. Next we have the mRNA vaccine. So mRNA vaccine is very famous one now, you know, because of the COVID-19. So the RNA material in the mRNA vaccine instructs our body to make a specific protein that is specific to the virus but does not harm the receiver when it is administered. The antibodies that can recognize the protein are created as a result of an immune response to the protein. In this way, if a person were ever exposed to the virus, the body would prefer to have the antibodies to combat it. Now the concept is same for every vaccine, you can understand. So here we actually use the insoluble nanoparticle capsule to like coat this specific gene, this specific part. So this is the entire genome, and this part is actually the specific part that codes for a particular antigen, and we coat it with the insoluble nanoparticle capsule. So here is the illustration. So we introduce the nanoparticle coated vaccine in our body, and this capsule, it fuses with the cell membrane and release the genetic code, and the cell's machinery, they will read this genetic code and will produce the viral proteins. Now these viral proteins, they are presented by the cell, and these antigens, they will be detected by the T helper cells, which will release the cytokines, and then the B cells will produce the antibodies, and also the memory cells are also created, so that when the body will actually face the attack of the real viruses, they can recognize it and can destroy it. Now here are some steps of vaccine production. So these are actually given in a very simple way. So we actually select the strain of the bacteria or virus that we need for the production of vaccine. Then we culture them, we harvest and purify them. Next we inactivate or attenuate the microorganism depending on the need, and then we formulate the vaccine by adding preservatives. And lastly, we have quality control and packaging. So these are some these steps are given in a very broad spectrum, so for the idea, just for the idea. And now we will discuss about how we can use egg in vaccine production. So recently we used the cell culture for the production of vaccine, but before that we used egg for the culturing of the cells for vaccine production. But this use of egg is actually beneficial because it has the excellent production capacity that is capable of producing an estimated 1.5 billion doses annually, and low production cost that allows global access to the vaccine. So for these two reasons, using egg is still a very relevant thing in vaccine production. And usually we prefer to grow the influenza vaccine in the egg for the vaccine production. So here are the steps. So at first we need an egg that is totally pathogen free, and we can use it after 11 to 12 days of fertilization. Next, what we do, we place a light source in front of the egg in order to locate the non-veined area of the allantoic cavity below the air sac. So you see the structure. So this area, this white area, is the air sac, and just below it we have this allantoic cavity which is filled with the allantoic fluid, and this one is the embryo. And using this torch, like see here, so this person is actually pointing the torch at the air sac, so this part is very light colored, and after that we have these red regions, this may be the embryo, and we actually locate the allantoic cavity which has no veins in it because it's a fluid. So once it is located, a hole is drilled at the top of the egg using the Dremel motorized tool. So this is the tool that we use, and we drill it here, like see this part, this point where I'm pointing. So we drill it, and next we use the tuberculin syringe. So this is a tuberculin syringe, and we introduce this syringe to inoculate the virus in the allantoic cavity. So the needle need to pass through this chorioallantoic membrane. Now this membrane between the air sac and the allantoic cavity is the chorioallantoic membrane. So the syringe, using the syringe, we need to introduce the virus in this region, in this gray region. We need to introduce our virus. So once it is introduced, the hole in the shell is sealed with melted paraffin or with hot glue gun, and then we will incubate the eggs at 37 degrees centigrade for 2 days. Now depending on the type of virus we are working with, we will incubate it in that way, and after incubation the eggs will be placed at 4 degrees centigrade to kill the embryo. So during the incubation period, the virus replicates in the egg, in the cells that make up the chorioallantoic membrane, As new virus particles are produced by budding, they are released into the allantoic fluid. So this allantoic fluid will be filled with the influenza virus. To harvest the virus, the part covering the air sac is removed. So this part, see this picture, this black and white picture. So this is the part covering the air sac. We need to remove this part, and very carefully we need to do it so that we don't disturb this allantoic fluid or the allantoic cavity. Now we use special tool for obviously to remove this part in a very neat way. Now the shell membrane and the chorioallantoic membrane are pierced with a pipette, which is then used to remove the allantoic fluid. So this step is we need to be very careful while doing it, because we just need the fluid, which is mildly yellowish fluid. We need to take it out because it is filled with the influenza virus, and we get about 10 ml per egg, and sufficient virus may be produced in 1 or 2 eggs to produce 15 microgram dose of vaccine. So this is how we actually produce the influenza virus that can be used for vaccination. Next, we have a cell-based vaccine production. So in this type of vaccine production, we actually bulk produce the virus in a fermenter, which has the following parameters like controlling of temperature, pH, oxygen, like that. And this mass production of the cell is done by two methods, like either by the microcarrier cultures or the free cell suspension culture. Now at first we'll talk about the microcarrier ones. So in microcarrier system we use a microbead. So this white one is a microbead, and we place these viruses. Let's say these are the virus. So we place these viruses on top of this microbead uniformly, and then this microbead is placed in a fermenter where this viral particle will grow, and something like this we will see. These microbeads provide a very high surface area to volume ratio, which can lead to high cell densities. Once a bead is covered, the cells are dislodged, dispersed, and allowed to reattach to achieve another round of cell growth on the surface of the bead. So once this bead is covered, we will take out all the viruses, and then this bead is ready for reusing. So this is how the microcarrier system will work. Next, we have the free cell suspension cell culture, where the cells the cells will be multiplied freely in a nutrient media, and we don't need any carrier for this. And the advantage of this type of system is that they can be expanded more easily, and the volume is also unrestricted. So this is how we mass produce the cells or the virus. Now next step is formulation. So formulation is the process of mixing the product, settling dose requirements, the concentrations and the final volumes, and establishing secondary process parameters like the materials, vials, syringes, caps, what are required for a vaccine. So after formulation, the next step is filling, and lastly packaging. So this is how the vaccines are actually produced. And that is it for today. Thank you for watching, and subscribe for more updates.